I'm going to talk about our research, mostly done at Hacettepe University. So I try to put the parts that we have done in Turkey. I accidentally said 2017, but actually I started in 2019. So most of the things are very recent. Some of them are unpublished. So hopefully soon we will be publishing many of the results that I will be sharing with you today. So our research mainly focuses on multisensory integration. In engineering, we generally use the term sensor fusion, but when we talk about animals, it's much better to say multisensory integration. So multisensory integration is the process where our brain, basically the central nervous system, combines information captured from different sensory systems. So imagine how much information you constantly get from the environment, like visual information, hearing, audio information, haptic information, and then even without noticing, your brain automatically combines them and makes some decisions for your for the tasks, whatever you are currently working on. The problem is, although this is so ubiquitous across animal taxa, we don't really know how brain achieves this sensor fusion task. So how what kind of filtering mechanisms applied, what kind of algorithms are embedded in your multisensory integration scheme so that you can successfully combine information you're constantly getting from different sensory systems. So the major problem that we will be focusing on is multisensory integration, but actually we extend this a little bit to multisensory behavioral control because we don't really study sensory systems as open loop passive sensors, but instead we study them linked to a plant in a closed loop control system. So whatever sensory systems we have on our bodies are actually intact with a plant, with a locomotor system that automatically reshapes the information you get from the environment. So imagine the optic information you get, and just think about how much optic shift you can create by just moving your head around. So whatever motor movements you do automatically changes the sensory signals you get from the environment. So sometimes this may seem like a disadvantage, but actually animals use this this closed loop nature in a very interesting fashion, where they can intentionally generate some useless motor actions. What I mean by useless is they don't really serve a motor purpose, but actually they change the sensory information you get from the environment. So most of the animals use unnecessary motor actions to reshape the sensory information they can get from the environment. So whenever we study multisensory integration, we automatically embed the term active sensing, which is the expenditure of, which can be defined as the expenditure of energy for the purpose of sensing. So what kind of motor movements we can do in order to reshape the sensory information you get from the environment. So this active sensing is also very common across animal taxa. One of the most known example is the echolocation in bats, where bats transmit sound waves into their environment, and then they use the reflections to understand what's going on in their environment. So they can change the pitch rate, the frequency of these sound waves, so that in different situations they can increase the accuracy of the sensory information they are obtaining from the environment. I'm not sure if the video is clear, but here there's a rat. Rats use active sensing a lot. They whenever they see any food, let's say cheese, it may be a trap. So they move their whiskers on that food to stimulate the smell. So when they get the smell, they can understand if it's something poisonous or if this is something they can eat. And as humans, we do many different types of active sensing. Even when you are choosing a watermelon, the step you do on the watermelon is to hear the sound, to assess the quality. So this is a nice example of active sensing you do. That motor purpose, that motor movement is useless, except it's helping you to hear the sound. You understand, assess the quality of the watermelon when you're choosing something. There will be some different parts. So I will first focus on our study on beaked electric fish. So how we use some animal models to understand how they're achieving this multisensory integration. Firstly, I will introduce our test animal. This is a Agameniurescens, also known as the glass knifefish. This is a weak electric fish. The names come from its famous electric organ. It's not like eels, where they use as like a to attack other animals, but instead here electric organ is only used for the purpose of sensing. I will show you soon. There are more than 200 species, but none of them lives in our country, so they're all wild catch. So whenever we work with this animal, we need to import them from Amazonian habitats like Colombia, Ecuador, from those countries. So this fish has very nice, unique features that allows us to use control theoretic framework to study multisensory behavioral control. First of all, they have vision, but the issue is they live in very murky waters with very limited line of sight. Basically, vision doesn't really help. Some of the species of electric fish actually lives in caves where there is no visual information. So some of them completely lost their visual system. So vision for this animal is not as strong as you may think in another animal. So as a solution to this problem, they evolved an electrosensory system. So here they have a long electric organ, electrosensory lateral line, where they constantly transmit an electric field. It works like a constant current source. And then the returning fields are captured by many electroreceptors distributed everywhere in their bodies, mostly in their heads. So using this electric field reflections, they can estimate the position, size, conductivity of the objects surrounding them. So even in dark, they have a nice representation of the environment. They can sense what is going on in their environment. This behavior also studied in communication theory. Like a lot of people, like there's another research area on how information is shared between individuals with this electrosensory information. But I'm not going to talk about that part. If you're interested in, there's also a part in communication theory working on this fish. In addition to these sensor systems, this fish has a very strong maneuverability, maneuverability. So normally there's a small anal fin at the back of the fish. So these anal fins are very important for the fish because each fin ray is controlled by 3 muscles. So they have a very strong maneuverability on that specific muscle network. But in this fish, that's normally small anal fin that you will see here is extended all the way down to almost to their mouth. So it's covering the entire bottom part of its body. And on this long fin, anal fin, they have a very accurate control so that they can do very different movements that most of the fish can't do. So they can just hover in place, they can move upward, they can swing backward. So it's something they can easily do that is impossible for many of the fish species. So another nice thing in the swimming behavior of this fish is along this long anal fin, they create 2 counter-propagating waves. So they initiate 2 different waves starting from different sides of their fin, like in the simulation. And then with these 2 waves, they can apply thrust either in forward or backward direction so that they can swim equally well in forward or backward direction. So in a single linear dimension, they can do sinusoidal type movements. And besides those, one of the most important reasons why we study this fish is they have a very important natural behavior, shelter-seeking behavior. So whenever you put an obstacle into the water, they automatically go inside that obstacle, and if you move it, they will track their movement. So here you can see this is a video from our laboratory. Like you can see there's no training for this behavior. You don't need to do anything. Just catch a new fish, put inside this aquarium. It will do exactly the same thing. So it will track your movements very well in that single linear dimension. And this is one of the fundamental reasons that that enables us to use control theoretic approach along this very simple one-dimensional linear tracking behavior. So we developed an experimental system, actually multiple experimental systems that I will show you soon, around this unique behavior. So here we create a flow tunnel. I will show you the mechanics of that part. So what we do is, instead of trying to move that refuge with our hands, we connect it to a linear actuator so that we can move it in any predefined trajectory you want. So you can apply constant frequency sinusoidal signals, you can do chirp signals, noisy movements, so whatever you can imagine now, we can stimulate the fish with that type of refuge movement, and with the camera system below, you can record the response of the fish. So this is how we are currently studying the behavioral response of this fish. And when you have this ability, you can do, for example, a simple frequency response function identification. So we can move this refuge in different frequencies. This is a movement at 0.10 hertz. It's very slow, but this video is captured from below again. So here, from this moment, we can track the response of the fish. So we can estimate the change in the amplitude. We can find the change in the phase so that we can create this frequency response functions in different frequencies. And we're generally working up to 2 hertz, where we are still seeing a significant tracking response. But beyond that frequency, fish doesn't really respond. It's very high frequency movement, and we don't really sense. We can't really say if there's a tracking or not. But up to this 2 hertz range, we are getting a very nice tracking response from this fish. So this is just an illustration of the mechanics of the experimental system. This is the design, 3D design. Here we have a thruster. This is Blue Robotics T200 thruster. So this is the source of the flow that we have in the system. So our goal was to imitate the actual Amazonian habitat where they swim against the flow. So we are also studying how the responses are changing when you are changing the flow rate. And then we have some guides. We have a honeycomb filter here to obtain a laminar flow in this test area. And this is where we actually place the fish. So this is just right here. So somewhere here. And then again, the water works in a cycle so that we have a flow tunnel here. And we do light and dark experiments. This is why like these black ones are infrared lighting. So we do experiments in complete darkness. As I said, only using the electrosensory information, fish just repeats the same thing. Right? It can track the movements of the refuge again. So in order to get those recordings, we use infrared lighting, and our cameras are also NIR cameras so that we can capture fish movement even in complete darkness. This is the electronics. All this experimental setup, mechanics, electronics, and software, everything is developed here in Turkey. Of course, with contributions of many graduate students, I will show at the end. So the main as the main computer, we are using NVIDIA Jetson NX card. I'm sure many of you know this. Maybe you already use this. This is like an improved microcontroller. I can say like there's a GPU inside, embedded GPU, and you can utilize GPU with the CPU together. Normally we have Ubuntu inside, but we are working with robot operating systems. So everything, all the programming is done in robot operating system. We have an NIR camera from Bastler, and then we have Maxon motor driver set. We have infrared laser arrays, and all the programming is mainly done in either C or Python in the ROS environment. So we are trying to obtain a real-time system as best as we can. So it's still, of course, soft real-time, but we are trying to, so for example, we are recompiling the kernel to change the operating system priorities, doing all of those to prioritize this task that we have in this in our experimental setup. So this is just an illustration of what kind of nodes operations we are conducting. So we have an image capture node, we have video recording, image processing nodes. So most of the things like video recordings are done online and in real time, so that we can use the movement of the fish as an input back to the stimulation signal. So this is an important part for us. So we are trying to improve this in different settings to get a real-time system as best as we can. Everything is running at 25 Hertz for now. So this is just a sample refuge tracking experiment. So just to show you what kind of data we are dealing with. This red one is a sum of science type input stimulus, which consists of 13 different frequency sinusoids. So it's a highly complex signal. And this blue one is the tracking response of the fish. Here you can see how well fish tracks it, like this multi-frequency signal. And this is just, again, a video from below. This is actual fish. We are adjusting the camera settings so that we see only the fish and the refuge movements to make it easy for in the image processing part. And this is just a summary. I'm not sure if it's the same data, but it's one of the sample data we have. The key part here is, if you look at the input-output frequency spectrum, you will understand the actual tracking behavior, like the BT in the tracking response of this fish. So these red peaks are the frequencies where we apply the input, where we actually move the refuge. It's all combined in a single sum of science type trajectory here, this red one. And if you look at the response of the fish, all the major peaks of the fish occurs right at the input frequency. So fish is responding to each and every frequency you see here. It's so nice that that in a single experimental trial you can get and you can estimate that you can get an idea of the tracking response of the fish. This is very this really simplifies all the analysis you can do, because you don't need to repeat different frequency experiments. It holds the superposition principle, so it's very important for us, and it makes our lives very easy to apply system identification techniques or control theoretic approach on the sample. But the problem here is this is an input-output response. So basically, whatever you estimate from here is an estimate of this closed-loop tracking response. So at this point, the only thing we can estimate is the input-output behavior from the refuge movement to the fish movement. So if you want to go inside this loop, so if you want to understand how the sensory-motor system is working or how the locomotor plan is implemented, so you need some interim signals, like something between a controller and plan, so that you can break this loop and get an idea about what is really going on inside the system. If you do this in other animals, like in humans, a common approach is to use EMG measurement or EEG measurement. All of these approaches are quite difficult in a free swimming fish. In our experiments, fish is completely free to do whatever it wants. So we don't force it to do anything. It's just their natural instinctive behavior. So in that free swimming case, it's really difficult to use other types of measurements. So we generally focused on two different approaches. I will show you the second one at the end of my presentation. But for the first approach, what we have done was to use a neuromechanical dynamic model for the locomotor systems of this fish. So this was a previously published model in the literature in PNAS. So this is just basic. It's very similar to a double integrator system, just modeling the frictions and some other phenomenon for a body swimming against the water, against the flow. So this plant model was tested in a very certain scenario against the flow, but it wasn't verified with the actual data from this fish. If you remember at the beginning of my presentation, I said fish uses this counter-propagating waves, just generating these counter-propagating waves to generate thrust in forward or backward direction. So what we have done was to use this data, the location of this nodal point where those two waves meet, in order to estimate the parameters of this model so that we can actually use it in our experiment. So if we can estimate this plan, if we can estimate these parameters, what we can simply do is, when you know the closed-loop transfer function from R to Y, and if you know P of S, it will be just a simple calculation to get an idea about the controller in the system. So what we have done was to record high-frequency videos of the movements of the fin of this fish during refuge tracking. These videos are stabilized according to fish. So actually, it's swimming forward and backward, but I just stabilize it to show you how the fins are actually moving. Here in the inset, I'm not sure if you're seeing below, but so here you can see how two waves are forming, and then it's meeting at a nodal point. So what we do is basically we digitize that point, that entire fin movements, and then we estimate the location of that meeting point, and we use it as a proxy of the controller output of this fish. So we know that this is an approximation of like the thrust information that fish uses to move forward or backward. So we digitize this fin movement and estimate that nodal point to use it just like an EMG signal or an EEG signal. We use it as a proxy of the output of the neural controller. So this is just a sample data at tracking from 205 Hz. So here you can see how the peaks and troughs of the fin is moving along time. And then this is the nodal point where they meet. So it basically, the nodal point here is forming a sinusoidal motion right at the input frequency. So there's a nice linearity between the nodal point movement and the fish velocity. So here you can see the correlation between the fish velocity and the nodal point movements. So we repeat this experiment in different frequencies. We can obtain multiple trials at for the same experiment to get an average response. So here this red one is the stimulus, and the gray ones, if you can see, are results of the fish from individual trials, and the blue one is the average response. And here the same thing, like for the nodal point movements, the gray ones are individual trials, and the green one is the actual trial. It's not really perfect sinusoid, but we try to estimate the sinusoidal function out of this movement, and then by using this data, we estimate the parameters of this previously established plant model. So when you have this plant model, as I said, so it was you basically you can get a frequency response function estimation for the plant. But most importantly, since we already know the closed-loop transfer function, now we can back calculate what the controller is. So we can simply calculate in this closed-loop system. We can calculate what the controller will look like given the HS, the input-output transfer function, and the plant dynamics. And here, what we have seen was this sensor-motor system has a high-pass filtering behavior. This was very important for us for our active sensing studies that I won't currently talk about, but this is something very important for us, just in the sense that the sensor motor system, that a neural controller, is weighting more on the high frequency information than the low frequency information. So this is something important for the details of our active sensing studies. So what we are currently doing is all the previous analysis I showed you was manually clicked. So we digitize fin movements by hand clicking on each and every frame. It was a very long process, but it was very important data for us. But here at Hacettepe now, what we do is to use a two-step machine learning approach to estimate that fin and then the nodal point. Maybe there's a slowed version. So here, what we do is we use a deep learning-based tool called Deep Lab Cut to first digitize the entire fin movements, and then we extract that fin movements and use it in a second stage machine learning tool in an LSTM structure to detect where the nodal point is. So here we only use the fin movements to estimate where the nodal point is. So this this is very important for us to generalize this this solution to different experimental settings, especially all the previous results we had was with constant frequency signals, simple sinusoid. But now we are using multi-frequency sum of signs type signal again. And then even in that case, we can estimate the nodal point very well in an automatic fashion. So, and the nice thing here is, again, I want to show you this red signal is the velocity of the fish. It's just negated so that you can see the correlation clearly. And blue one is the movement of the nodal point. Here you can see how linearly correlated, how well it's correlated with the velocity of the fish. And now by using this information, we can estimate the plant in a single experiment with a sum of signs type input signal, and then we can switch to the calculation of the sensory motor transformation. For the multisensory integration part, so how to connect all these results to multisensory integration, we first do a comprehensive analysis on the sensory salience. So the issue here was during all of these experiments, fish automatically combines the visual and electrosensory information, and we don't know how it's weighting them, how it's adapting itself to different scenarios. So what we did was to change the sensory salience. Salience here means the ability, or it's like a metric of how well a sensory cue stands out from surroundings. So how clear, how distinguishable it is from its environment. So, for example, for a visual cue, for a visual signal, changing the light automatically changes the salience of the sensory information. It becomes much more difficult for you to detect the edge or estimate your state in a dark environment as compared to a light environment. For this fish, we changed the illumination for the visual cues, but for the electrosensory cues, we changed the conductivity of the water by adding a salt mix so that we can modify the performance of the electrosensation system. And we're also changing the structure of the refuge. So again, both the visual information and electrosensory information will be affected by the change in the length and by adding some windows or like no window case for this fish. And we started testing how the behavior of these fish, we are currently using 3 species, are changing as we are changing the salience of the sensory information. We're also testing with flow speeds. I won't really talk about that part for now. So that flow speed is important for us to understand the contributions of the mechanosensory systems of this fish. So mechanosensory system is, they have some very specialized cells. Actually, we also have them, but it's not as strong as the fish. So with the mechanosensory system, most of the fish actually can feel the speed and the direction of the flow. So that's something, topic of different study. So our initial results here with the sensory salience experiments were really important for us. So this histogram shows us the variability in the behavioral output of the fish under different sensory salience conditions. So here, these are light experiments, dark, like refuge with windows and no windows, and this is just the colors are indicating the length of the refuge. Here you can see how, for example, how the behavioral variability of the fish is increasing as you're increasing the length of the refuge. So, or if you're switching from light to dark, you can see the dramatic change in the behavioral variability of the fish. So this behavioral variability, you can think this as a measure of consistency in the tracking response of the fish. So basically, when the sensory salience is well, so when the information is very clear, it's easier for the fish to do state estimation and perform the tracking behavior well. And when you repeat this, it's very consistent. But if you switch, if you go through low salience regimes, then automatically the variability in the behavior of the fish increases. So this is important for us in different sense. So this is critically important to study the functions of the multisensory integration. So there are different roles of different reasons why our brains are doing multisensory integration, and one of which is to increase the reliability of the information. So basically to increase the consistency in the behavioral outputs. The other metrics are generally to speed up information processing later on in different results. We are seeing how the phase lag is changing as you increase the sensory salience. So this was one of our initial results that we were able to publish up to now. So all the results I showed were in a scenario where we are moving the refuge, and that refuge was concomitantly like transmitting visual and electrosensory information. The issue up to this part was we were really able to estimate vacuous response functions of the tracking behavior or plant dynamics. We can calculate the sensory motor system, but the problem was we can't really distinguish the contributions of visual signals from the electrosensory signals because they're originating from the same source, and we can't really identify how much visual lies on one of them. And for the variability, it will be for our recent results. We can let me show you this person, then you will understand that part. It's so for this part, what we tried to do was to distinguish the contributions of visual and electrosensory signals. So what we did is to find a way where we can actually distinguish the contribution, distinguish the signals for the visual and electrosensory part. This was the first initial design we did. This was inspired by, again, a recent study in the literature. So the idea here was to use a translucent refuge where it's like a yarı saydam. So it just passes some part of the light, and then if you have a projector here, and if you project light stripe just like the windows, so what you basically do is if you move them together, it will be similar to what we have done in the other experiments. So the movement of the refuge will provide the electrosensory cues, and since the projector is attached to the same linear actuator, it will move together in synchrony. So the movement of the light stripe will create the visual cues. So this will be a very similar experiment with the other case. But the nice thing here is now we can distinguish, we can separate the visual information from the electrosensory information however we want, because now, for example, we can all these experiments are in dark, by the way, so we can keep the refuge, the actual physical refuge, stationary, but only move the light stripes, which will basically stimulate the visual system, but there will be no electrosensory information. Or we can do other ways. Like we can move the actual refuge, create the electrosensory information, but we can keep the light stripes fixed. So that was the first thing we tested, but the problem here was I will show you from our results. So when you have those light stripes, the whole logic is fish shouldn't see the movements of the actual refuge, but it wasn't really possible for us to eliminate that sense. So even with our own eyes, in different parametric settings, it was still visible, like the movements of the actual fish, this gray part were still visible for the fish. So we tested with very special color filters, like color filter lens, but they didn't really work well. So we gave up on this design, but instead we completely redesigned the system to use two different actuator systems together in this for the same refuge. So here we have a highly transparent plexiglass refuge inside. So the movements of that refuge will provide electrosensory information. And on top of this, we have a 3D printed, basically, refuge, which has embedded neon lights that works underwater to provide the visual cues. And we tested this many times so that the movements of this outer tube doesn't really create any electrosensory information because it's very short range, and this inner plexiglass tube blocks the electrosensory signals, their range as well. So what we do here is we have two different actuators. They can move in synchrony, or they can move completely with a different setting, so that when they move in synchrony, it's going to be the same experiment with what we have done before, but actually in this new system, what we can do is we can separate the visual cues from the electrosensory cues, however we want. Now, we have complete control on the trajectories of visual and electrosensory information. So this is just a rendered video. I hope it's clear. So just to show you how it looks from inside, so what the fish is experiencing. So it has this embedded neon lights. So this is the current setup that we are using. So we are currently testing the response of the fish to an smoothly edit frequency content. So what we are trying to do is to measure the weights of assigned to visual and electrosensory information in an unconscious manner. Otherwise, if fish really understand what is going on, if there is a sensory conflict, sometimes they don't like to stay inside the tube. They're getting nervous. So we try to smoothly add a third frequency component to see how unconsciously its behavior it's responding to this change in the sensory information. So here I will try to explain this as best as I can. So this is a 1 minute long experiment. In this first 25 seconds, here the red one is electrosensory system, the trajectory of the inner tube, and green one is the visual system. This is the trajectory of the outer tube. So in this first 25 seconds, and here you can see the frequency response in the signal in frequency domain. Here it has only two frequency components. So red and green are only appearing here at 0.1 hertz and at 0.35 hertz. The blue ones are the movements of the fish, and this is the response of the fish. And again, this is the response in the frequency spectrum. And then here in this 10 seconds, we smoothly add a third frequency component, I think in this case, to the electrosensory system. So as you can see in this 10 seconds, the trajectories of the red and green curves are separating. So now separated, here we are adding a third frequency component to the red one. So and then in this last 25 seconds, we have three frequencies for the electrosensory system, but two frequency stimulus for the visual system. And this is the frequency response. And you can see again, at 0.1 hertz and 0.35 hertz, they're identical, but at 0.25 hertz, we only have a component in the electrosense, but not in the visual system. So our goal here is to do this by only adding this third frequency component to an electrosensory stimulus, and then we repeat the same thing by adding the third component to visual stimulus. And then we do the same thing by adding this third component to both stimulus. So by using these three plots, we can identify the weights assigned to visual and electrosensory system for that specific frequency. And of course, we can change this frequency, and we can try to estimate the weights assigned to each sensory stimulus at different frequencies. So by taking the ratios of the gains in this so-called visual response at 0.25 hertz or this electrosensory response, we can try to identify how much weight is assigned to different sensory systems, so how much they contribute to overall behavior. So as you can see in the electrosensory case, the gain is quite high, but not in the visual system. This is something normally we weren't expecting, because we always believe that visual systems is much more stronger. And we still agree on that part, because when they are working together, we still believe that visual system dominates the electrosensory system. But remember, in these two scenarios where we had sensory conflict case. So basically, when you have visual information here moving outside, the electrosensory system inside the transparent refuge inside is not stimulating the electroreceptors at that frequency. So we believe that electroreceptors have key importance in stimulating the movement at a specific frequency. But when they are combined, visual information is more dominant. So when there is no sensory conflict, we believe that visual information is more important. But electrosensory input alone visual signal alone is not as strong as the electrosensory case, and here it's not really, the response here is not really distinguishable from its surroundings. So the signal to noise ratio is not something significant to assess. We use them, we study them in different settings. So we try to estimate the gains in different scenarios, not gains, the complex functions, I should say. So this GEV is when both electrosensory and visual information are present. Here, this star is the ideal tracking case when the gain is 1, basically the amplitudes match, and phase is 0 when there is no phase lag between the two input-output signals. So this is the perfect scenario. Here you can see GEV is very close to 1. And if you look at the electrosensory, its gain is very close to 1, but it has a huge phase lag. Visual system, its gain is low, and there's a huge phase lag again. So here, and the diameters here are not really that correct. I mean, this was the diameters here comes from variability in time domain, but now actually we are trying to do a variability in complex domain, so that we have ellipses in like point in different directions. But again, here the idea was when we were now with the frequency with the variability in frequency domain, what we see is when both information are present, the variability is generally much reduced. So it's again, we are seeing the effect of sensory integration and consistency. But for now, the diameters here is not representing the frequency results, but they're coming from the variability in time domain. Any questions up to now? I don't know how much time I have, but I was going to talk about the zebrafish part. I can quickly, I can completely skip it, or I can quickly explain what we do with zebrafish. So we're also working with another species to generalize this to different species, different animals, so that we can get an idea about what really happens across species and within the same species. So I will, without explaining in detail, I will try to quickly show you what we are designing for the zebrafish. So if you know zebrafish, they're really like new lab rats. It's used everywhere now. It's very easy to study. So instead of experimenting with rats, people really prefer to work with zebrafish. So zebrafish has a very nice, very important behavior called rheotaxis. So this is a natural thing. Again, you will see rheotaxis is the intentional orientation of body toward the flow, so that they can achieve station keeping against the flow with minimal energy. So when there's flow, so when you turn on the flow, these fish quickly turn their bodies toward the flow so that they can do station keeping with minimal energy. So our idea here was to utilize this, again, natural behavior. So if the sole goal is to minimize the energy consumption, then what we thought was, let's add an obstacle to obscure the flow in a certain region. So if the goal is to minimize the energy, that fish should go behind that obstacle. And we can take this to the next step. So if you move that obstacle, this time in horizontal direction, now the fish should track its movements if its goal is to minimize the energy. So when there's an obstacle here, there are two, again, dominant sensory systems zebrafish use. So they have visual systems, so they can see the location of the obstacle. But most importantly, zebrafish has an extremely strong sense of mechanosensory information. They can accurately estimate the speed and direction of the flow. So they are very good at this. So whenever you add an obstacle, it will change the gradient, the flow gradient in their environment. So they can, even in dark, they can understand where there is low flow speed and where this flow speed is higher, so that they can position themselves behind those obstacles without even seeing them. So we developed this experimental setup. It's very similar to the one I showed with the weakly electric fish. This time the movement is in the horizontal direction. I will set up. It has the same nature, same electronics, and same software technically. Just a small requirement for the zebrafish. And I will just show you a sample result, just a case. So just to show you how fish is responding to these movements. We're moving that tube, which is obscuring the flow. And this is again a video recording from below. And we are tracking the movement of the zebrafish, the fish and the tube that we are moving. Currently, we only do either a constant frequency movement or two frequency content. We weren't able to do a multiple frequency case for zebrafish. They're not really good at tracking at high frequencies. Good at tracking at multiple frequencies when they're together. So just to give you an idea of what we do is, so again, when this tube moves, it concomitantly transmits visual and mechanosensory information at the same time. So they're originating from the same source without any conflict. But in order to get an analog version of what we do with the electric fish, what we do is we use a completely transparent outer tube, so which provides the mechanosensory information. But inside that tube, we place either again a neon LED or an handmade mimic zebrafish, just a copy, or just a colorless stick. We are still testing which one is more dominant to create a visual information for the zebrafish, and we are testing this under different scenarios. So what really happens here is, again, we have maybe here in the design. Oops. It's waiting for the video. So again, we have two actuator systems. So one is moving the outer tube, one is moving the inner visual object, so that we can either move them in synchrony to get to do the same thing with the weak electric fish, or we can move the inner, just the visual object inside the outer tube, or we can do other ways around to estimate the contributions of visual and mechanosensory information. So I will skip all of those, just a simple tracking result in light and dark. Here, mechanosensory information is so dominant for this fish, so that even in darkness, we are still seeing very nice tracking response, just different. And again, we are estimating this these cases. I will skip all of those. So I will talk about two research directions we have. So this is also just to advertise our lab to everyone here. If you would like to pursue a graduate degree in these topics. So in the first part, we are going ahead to study the neural mechanisms of multisensory behavioral control. So here what we do is, with the same experimental setup, we implant electrodes directly to the midbrain of the fish, so that we can get a neural recording of the visual neurons, electrosensory neurons, and where in the optic technique where these things are combined, so that we can study multisensory integration in the neural level. This is something I have done at Johns Hopkins University, but now we are trying to transfer that experimental system to here. And second part, I am sure all of you know this robot from here, and so we are trying to translate this knowledge to novel sensor fusion techniques for robotic applications, so that we can use them, we can compare them against cutting-edge sensor fusion tools in engineering. So is there any novelty, is there anything we can bring back from biology back to engineering, so that we can develop new sensor fusion algorithms for robotic applications? This is just the funding support we have here. All of these things are supported by many sources, especially from Mercury Actions from European Union. And of course, all of these things were only possible with a swarm of people, like one postdoc, currently 8 graduate students, full-time graduate students, and many undergraduates that are helping to build all of these things possible. Thank you very much for listening. I will be happy if you have any questions. Happy to answer.